Hiba and Atif. Hiba, can you speak today? Would you like to give me a brief summary of the last class? Hiba, are you there? Or is it like, I think from a very long time now, me and Atif are just doing this. Hiba, if you can do this time, it will be great. Were you there in the last class, Hiba? Where did we start the last class, Atif? No? I think this was done.
We started translation.
Transcription, right?
Translation.
Translation. Just one moment. This is MEC. Oh, sorry. This is MEC. Oh, yes. Sorry, gets confused in between the batches. Oh, we are way ahead in syllabus then. We did translation already. Did we finish complete translation? Gene expression, operon. Oh, that's great. Okay, very well then. So today we have to do human genome project, correct? Probably we can finish the chapter today, I think. Okay, any doubts? So where did we start from? I think we start from translation. So Atif, I think it's again going to be you and me doing the talking in the class. So would you like to begin? As always.
Sir, actually, I have a fever and I have a sore throat, so I can't speak for long periods of time. Can you
Okay, so yes, so let me do it. So it's me and Atif. We have to do it on a rota basis. Please don't miss classes. Rifat, please get your microphone fixed. See, otherwise it's just me and Atif revising things. Okay, so let me start with the revision today. So we started with translation, and as we know that translation is the process of polymerization of amino acids to form a polypeptide chain, right? Just like transcription was the polymerization of nucleotides to form a RNA. Here it is amino acids. Now, two amino acids are joined together by a peptide bond, as I've told you. And then we went on to understand tRNA, which is an important crucial factor in translation. tRNA is called transfer RNA, or also it is known as the adapter molecule. So in a 2D, the books often describe tRNA as a L-shaped molecule, but actually in 3D, it's clove leaf shaped molecule. Okay. Now it is a long mRNA from 5', which has a 5' end and a 3' end. And good evening, Zed. So just how are you?
Fine.
I'm just revising it, okay?
Yes.
Starting with the revision. And this tRNA has at the 3' end, it has an amino acid binding region or amino acid acceptor region to which amino acids bind, or when they are charged, they get attached here. And it has an anticodon loop which reads the codon on the mRNA and brings the amino acid for which that codon codes for. Okay, so anticodon loop has a complementary sequence to that of the coding, like the sequence present on the mRNA. So tRNA is very specific for each amino acid. So one tRNA can only bind to one kind of amino acids. So there are 20 amino acids. So there are different separate tRNAs for each amino acids. Now I told you about the initiator tRNA. This is the specific tRNA that starts translation in any system. And as we know that what is the start codon? Start codon is AUG. So initiator tRNA contains methionine amino acid. Now charging of tRNA or amino acylation of tRNA is the process by which amino acids gets activated in the presence of ATP, and then they link or they bind to their specific tRNA. Okay. Now this was the process of translation that we discussed. So in the process of translation, as you can see, there are two sites, A and P, which are marked by red and blue here. in the ribosome's bigger subunit. The smaller subunit of ribosome binds to the mRNA, and then tRNA carrying a particular amino acid comes and sits at the A site first, reads the code, and then the ribosome shifts, and the tRNA shifts to the P site. And then a new tRNA comes and sits at the A site, and the tRNA which is at the P site transfers the polypeptide chain to the tRNA on the A site, forming a peptide bond between the two amino acids. And this continues till a long polypeptide chain is formed. And finally, when it reaches the end, where there is a UAC or any stop codon among the three, then no tRNA comes and binds. Instead, some proteins, which are known as terminator proteins, they sit, okay, and ends the translation, the process of translation, okay, also known as the release factor. So this protein is also called the release factor. Then we went on to study the gene expression regulation. Now, at how many different levels can you regulate the gene expression? In eukaryotes, we learned that there are many levels at which the gene expression can be regulated. First, the transcriptional level itself, whether you make mRNA or you don't make mRNA, is going to affect the translation directly. Second is the post-transcriptional processing level, because we know that the mRNA gets transcriptionally processed and modified with capping, tailing, and also splicing. So at that level also it can be done. Then the transport from the nuclear to cytoplasm. And finally, at the translational level, we can regulate the gene expression. Whereas in case of prokaryotes, there is only one way to regulate or control the rate of, to regulate the gene expression, that is to control the rate of transcriptional beginning. Like initiation should be regulated, because once it's initiated, transcription and translation is coupled, so it's difficult to regulate it after that. And then we discussed about the Lect system. Okay, we took an example of Lect operon, which is basically a cluster of genes under a common promoter. So these three genes, Z, Y, and A, that you can see here, are under this one particular promoter. And in operon system, there is one more component apart from promoter and the genes and the terminator, that is operator. Okay, and this operator region is where a repressor protein can come and bind and keep the whole cassette in a repressed state. Okay, and this repressor is also encoded by a gene which is just adjacent to the whole operon, to the whole cassette. Okay, and it also has its own promoter. So basically, this is called a negative regulation, where mRNA can sit at the promoter of the repressor gene, can make the repressor RNA, which can get converted into repressor protein, which then can go and bind to the operator region and will not allow any other gene to be transcribed further. Now, if you want to do transcription, if you want to do transcription and translation, then what you can do is, in the presence of an inducer. So inducers are molecules which are present in the surrounding, which gives a signal or a cue. So whenever these inducers are present, for example, in this case, it is the lactose. So if lactose is present in the environment, then these genes are required. Okay, so lactose goes and binds to the repressor itself, inactivates the repressor so that they can no longer go and bind to the operator. And hence, mRNA can sit at the promoter of the operon and can transcribe all the three genes as one single mRNA, because I told you it is polycistronic. And then when the protein will be formed, three different nucleotides will be formed from that mRNA. Is it clear, everyone? So in the presence of inducer, there will be a transcription and a translation happening. But in the absence of the inducer means something that induces a process. So here, transcription and translation are being induced. Everything is clear?
Yes.
Any doubt?
Yes.
No doubts?
No, sir.
Okay. So let's start with today's topic. And today we are going to begin with Human Genome Project. I think this is the next thing, right? So you can, before going forward, you can write down this lac operon is an example of negative regulation. So it is known as a negative regulation operon. Why negative? Because something is required to make it happen. Otherwise, it will always be in a shutdown state, because repressor will be in a bound state to the operator. Only when lactose is present around or any inducer is present around, then only this repressor protein will be inactive. So that is called negative regulation. The opposite of it will be a positive regulation. Okay, where the presence of something acts as a instead of a repressor, there is something called activator. So the presence of something acts as an activator or makes the activator transcribe and translate, and then the activator goes and binds and the process starts. So it's opposite, but mostly operons are under negative regulation, like operon. Okay, so now today let's start with Human Genome Project. So what does the word tells you about Human Genome Project? What do you think it is? People, yes.
Maybe like a project to research and know more about the human genome.
Yes, Human Genome Project was the first of its kind mega project in biology. It was the biggest project of that time that was undertaken or that was started in science, and it was a very, very ambitious project. And the project was to sequence the whole human genome. Now we are talking about, if even if you are taking a haploid, and of course we will be taking a haploid genome, because there is no point to sequence a diploid genome, because basically the genes are same. Yes or no?
Yes, sir.
So this haploid genome, even if it is haploid genome, human genome is, how big is human genome? We have discussed human genome. Any idea? And we are talking about haploid here. It is 3.3 into 10 raised to power 9 base pairs. So you can imagine at what scale we are talking about. So we have to sequence all of these bases to know what is the sequence of the whole human genome, starting from chromosome number 1 to chromosome number 23, okay, which is the X or the Y chromosome. So 22 autosomes and one sex chromosome, and both the sex chromosome, because X and Y are different. So we have to actually sequence around 24 chromosomes here. You understood why 24? 22 are same and 2 are different, right? So we have to sequence both to know. So, and this big, and at that point of, at that time, when this technique was being developed and was being used in this mega project, to sequence around one base pair, okay, it took around $3, $3. So that was the estimated cost for at the beginning. So even by going $3 per base pair, how much money do you need? Imagine.
Sir, by sequencing a base pair, what exactly did they do?
By sequencing a base pair, I mean you have to okay, let me first. So let's say this is a strand of the DNA. Okay. So has bases. If we sequence one strand, it is enough because we can know the sequence of the other, right? We don't have to sequence both the strands because they are complementary, correct?
Yes, sir.
So let's say this sequence is A, G, C, A, T, G, and so on. So what they actually did was to open the DNA, get one strand, and somehow by a technique, you have to get to know that which is this base at this position, and then this base, adenine, guanine, cytosine, then adenine, and you have to keep recording that in a computer. So that's called sequencing. Now we'll talk about sequencing in detail later on. How can we do that? But imagine 3 into 10 to the power 9 base pairs and sequencing it at the cost of $3 per base pair. It will be around more than, not around, it will be more than 9 billion US dollars. Can you imagine 9 billion US dollars? And this is all happening in 1990. Even today, so the project kick started in 1990, which is like how much? 32 years ago. So even today, this is a lot of money. So at that 32 years ago, 9 billion US dollars was the total funding for this project required. And it was funded. You know, the amazing thing is that it was funded, and this enormous amount of money along with enormous amount of data generation was, scientists thought that, okay, we'll do it. Now, why would you like to do such a thing? Why do you want to know the sequence of our genome? Now, today human genome is sequenced. Along with human genome, there are many other organisms whose genome are sequenced. But what will we get from sequencing genomes? What can you figure out if you can just keep knowing ATGC like till 3 billion base pairs without a comma or a full stop or something? You make a database of that. What is the use of it?
So you can like research can be done based on that. For example, like in the last chapter, you learned about characters and traits. Like with the current research, these can be associated with the base pairs, and more can be known about the DNA.
Yeah, what more? About the DNA, we can only know that DNA is made up of 3 things: phosphate, sugar, and bases. By knowing the sequence from the beginning till the end of all the 3 billion base pairs, what will be the benefit? Now that you know that how, what is start codon, what is stop codon, what can we find by this very long sequence? It's simple. We can find how many genes do we have, because we know how to track a gene. Every gene starts with methionine, AUG, and then ends with one of the stop codons, right? That is called the reading frame. Remember, I told you about the reading frame.
Yes.
So we didn't know how many genes humans have before we started this project, before scientists started this project. And they were estimating, because humans are the most evolved organisms on the planet. We are the most complex of the beings. You know, we used to think that before that. So we must be having lots and lots of gene for lots of things that we do. Like only for our biochemistry, like our metabolism, we must be having more genes than any other organism on the planet. But that was proven to be wrong. It was found that scientists were thinking that there is somewhere around, we will find approximately. So this was their hypothesis before this project started, that we can find somewhere around 1 to 1.5 lakh genes in humans. Such a big genome must be carrying 1 to 1.5 lakh genes. Do you know how many genes do we have? How many genes did we find? from the whole genome sequencing? Approximately 28,000. That's it. Surprised? Disappointed? They were very disappointed. We have just 28 to 29,000 genes in our body. Wait, let me tell you one more thing. This is for humans. You know how many genes do butterflies have? Some butterflies can have up to 33,000 to 35,000 genes. What does it mean? Humans have less genes than a butterfly? Bizarre, no? Waste of money. Then how are we special? Why are we special? We always hear this thing that humans are special. We are this, we are that, we are at the top of the chain. We don't even have genes. Like we have lesser genes as compared to a butterfly. So what do you think is happening here? Is it the genes? Is it the number of genes that decides complexity? So is a butterfly more
Sir, can I ask a
evolved than
question?
humans? Sorry?
Sir, can I ask a doubt?
Yes, please, Adil.
Like, where do they get all of these genes from?
In the DNA. They read all
Yes,
the,
sir.
they noted all the sequence. So if you starting from chromosome number 1, the sequence started with something, okay? And it kept going. And where chromosome number 1 finished, they put a stop, then started with chromosome number 2, again sequenced everything till the end of the chromosome, let's say the Y chromosome, okay? And where it ended, in between the first alphabet and the last alphabet, there were around 3 billion base pairs, right? Too
So
much
my
of
question
data.
is like, where did they get the genes from? Like, what was the source of these genes?
What was the source? Human DNA. You
Yes,
take
sir, but where
any
do they
cell,
get the human? Oh,
cheek cells. You take any cell, every cell has nucleus, right?
yes, sir.
Every cell of our body has a nucleus, and every nucleus has 46 chromosomes because we are diploid. Every cell is diploid. You can, if you want to take a haploid genome, you can take it from sperms or ovum. If you want to take it a diploid genome, you can take it from cheek cells, any nasal swab, anything, anywhere. You will just take a toothpick, scrap it in your cheek, and you will get some squamous epithelial cells and get the DNA out of those cells. Simple. Understood? Atif?
Yes, sir.
Yeah. So my question is a butterfly more evolved than humans? Because they have more genes? What do you think? Yes, no, nothing? Can it be more evolved than a human? Of course not. We are more evolved, right? But we have less genes. So clearly it is not just the number of genes that defines complexity. It's not like lower organisms will have less genes and higher organisms will have more genes in that proportion every time. So what is it that defines complexity? For that we will discuss some other time. But just know that Human Genome Project, write down. Let's define this project, write down. The Human Genome Project was a very ambitious project aimed to or aimed at. The Human Genome Project was a very ambitious mega project aimed at sequencing the whole human genome, which is approximately 3 into 10 raised to power 9 base pairs, which is approximately 3 into 10 raised to power 9 base pairs. Now, just to give you the context of how big was this project and how big was the data that was going to be generated, it is like if you start storing it in book, let's say, and it has been done also, I think. Let's say if you have to make a human genome book, okay, containing all the base pairs. So if every book, so say, so each page, if contains, has to contain around 1000 letters, okay, which means the alphabets, individual alphabets, ATG 1000 letters, okay, and then each book contains such 1000 pages. You understood? So each page has 1000 letters, each book has 1000 pages. You will require somewhere around 3300, sorry, got very ambitious, 3300 such books to store the human sequence. It's very big, right? Yes. Amazingly big, magnanimous. So this is the amount of data that we are talking about. Now write down. Human Genome Project led to the development of. So this kind of data, can only be worked on or can be accessed only through computers, right? If I give you if I give you access to a library which has 3300 books, so all the books are containing human genome, and suppose you are working on a gene and you want to find on which chromosome that gene is, will you be able to find it out? No, you have to go through 3300 books and reading each and every sequence. So the only way to do it, any kind of research on human genome, is through computers, where you can actually search things with a processor from those 9 billion base pairs, what you want, what you are interested in, and where it is present. So Human Genome Project was not just an ambitious project from biology point of view. It also led to, write down, Human Genome Project led to the development of a new area of biology. New area of biology called called bioinformatics. Bioinformatics. So this is biology done in silico. I told you about these three words: in vivo, in vitro, and in silico, right? So bioinformatics. You know what is in silico, remember? Computers. So now Mendel was the first to start using mathematics to explain biology, and many decades after, centuries after, now we are into the era where biology is being done through computers as well. And now even artificial intelligence is being used to do research in biology. Now, such a big project with an investment of more than $9 billion, there must be some big goals, right? And this is a CBSE question, board's question. Describe the goals of Human Genome Project. So write down. To identify, first goal, of course, was to identify total number of genes in humans. So this line, if you'll go to NC, and everyone has NCRT with you right now. I just found this, that if you go to your NCRT, it in one of the goals, it tells you that one of the goal was to identify all the 20,000 to 25,000 genes in human DNA. You know, it was not known that we have 25,000 genes. So that sentence is kind of, I don't know what it is. You know, you cannot just know beforehand that we have 25,000 genes and then go on to do this $9 billion project to figure out the gene sequence. How will you know how many genes do we have unless and until you have the whole sequence? You understand? Once we have the whole sequence, then we can find the number of genes, because genes have a characteristic feature. They start with a start codon and they end with a stop codon. In between is the reading frame, open reading frame. So that's how the genes were identified. So the aim, in fact, you should write, was to identify the total number of genes in human, and which turned out to be around 25 to 28,000. Some books write 20,000. 20,000 is way too less. We don't have 20,000 genes. We have approximate around 28,000 genes. The second aim was to determine the sequence, determine the complete sequence what is the complete sequence of around 3 billion base pairs. In human. Third goal was to store this information in the form of database. And even to store this as a database, you know, you need a huge, either and at that point of time, you know, servers kind of things were not there, like they were not the kind of cloud servers, so they have to do it in storage devices like hard drives or the actual storage of the computer. So database storage, which can be accessed. And once you have the data stored, then you can start doing whatever you want, because even scientists didn't know what all can we do with this data. Like Atif said, a lot can be done. You know, we can find out so many things from the data. So the data is like a Pandora box. You open that whole box, that see, this is the 3 billion base pairs in this sequence, and this is the sequence. After that, we can start a lot of things. We can find genes, we can try to cure diseases and everything. We can get to know about the nature of certain genetic disorders, their inheritance, etc., etc. What not can be done. So database storage, and then improve or develop tools for data analysis. Improve and develop tools for data analysis, because so big data can't be analyzed manually, right? Fifth was to provide the technology to industries so that industries can start coming up with different ideas, different like therapeutic things, medicinal things, gene editing tools, etc., etc. So provide related technologies. And this is where they will get, like they will earn, right? You have invested $9 billion, right? You need to do something good with it. Yes or no? If you just generate so big data and keep it close, is it of any use? No, right? You have to put it like in an open source, share it with different industries and companies who can come up with plans and ideas to work on it. So provide related technology to the industries, to industrial sectors. So fundamental science, whenever it finds something, we cannot, whatever we find, we cannot bring it to human use in a lab, right? We do the discoveries in the lab, but for it to reach the masses or for the greater benefit, we have to rely on industries, right? Scientific industries. Suppose a drug is found, a new drug has been discovered that cures a particular disease, but a lab or a scientist or a group of scientists in some university or research center must have discovered it. Yes or no? But who will put that into use when it goes through the drug trials and on the clinical trials and to come into the market? It is the industries that produces. Yes or no? You understand, people?
Yes.
Yeah. And lastly, when this thing was being done, whenever something new comes up in science, always from, like, you have to justify it to the society, because whose money was it? 9 billion US dollars. Whose money was that? Taxpayers. All the science that is done in all the countries around the globe, whose money do scientists use to do the science?
Government.
People, taxpayers' money. Where does government come brings money from? Government is not, just making money out of the blue. Money is what money does. Economics. If anyone has studied economics, the first line about money is what money does, right? So if that GDP, all these things, so we don't have to go there, but it's the taxpayers' money in short. People pay taxes, that money comes to government, then government decides how much to give to the research and development, how much to give to the construction of the roads, highways, universities, everything. So projects like this with such a huge investment done by the government, the ministries of technology or science or defense or whatever, they have to also address the ethical, legal, and social issues, right? That may arise from the project. So the last goal of this was to address, of course, that's going to be one of the goals, that how will they address, what will be the plan to make people believe that what they are doing is not ethically, legally, or socially wrong. Like it is nearly impossible to justify the ethical or social or legal issues with human cloning, right? Would you like human cloning to be a real thing?
No.
No, right? So no government can, just say that, okay, we give a green flag to human cloning, because they know that it can't be justified. And because the government setup, and it's not the monarchy rule where the king decides whatever has to be done has to be done. It has to be discussed with the people. So address the ethical, legal, and social issues. That may arise. from the project. Okay. So for the Human Genome Project, it was not just like one human who was chosen as the representative. In fact, there were many individuals chosen from different backgrounds, from different countries. You know?
So I just did like a Google search, and apparently the Human Genome Project was completely like finished in this year only, like in January of this year.
January of which year?
This year, 2022.
What was completed? What are you talking about?
The Human Genome Project was like complete, every base pair is like completely sequenced in January 2022.
It's not that Human Genome Project got completed in 2022. Human Genome Project got completed in 2006. You understand? Like, I don't know. What did you read about? Tell me. So as per your own textbook, which is NCERT, it got completed in 2003. So in 2003, the last of it was around 92% of the genome got covered, and they were sure that the rest 8% is likely to have some vital information. So we need to finish that also. But they took 3 more years to finish certain. So when something gets sequenced, it is not as easy that you can start from one end and like a zipper go to the other end. It was done in a very, very what do you say, tedious and hectic manner, because the data came from in between everywhere, and then they were arranged on the basis of complementarity and logic. Okay. So what you are talking about, maybe it's some extension work that they did because it got completed with respect to finding all the genes. It's not like that we just got to know today that in 2023. So whatever you are reading, just you can either share it with me. You have my WhatsApp information, then I can get back to you. Okay.
So it's the
Regarding that.
Wikipedia page, sir.
Yeah, and okay, and what does it say? Can you read it? Let me see what you're talking about.
So it says the final gapless assembly was finished in January 2022.
Okay, yes. So that is not a core part of the sequencing. That is the core part of arranging it in order to do some research that they wanted to do. It is still being done, and for the next 5 to 10 years, people will continue to work on the data to make it N. And one more thing, that a lot of new data is being added now, because now sequencing is not that expensive, and you can sequence faster and economical. So even the fossil records, and that we will discuss in evolution, it's very, very interesting. Just like we find fossil bones for animals, there are fossil records for humans also, how previous humans used to be like. So if we get, let's say, a skeleton of a human which dates back some 1 lakh years ago. Now 1 lakh year ago, Homo sapiens were not Homo sapiens as per, right? This species diverged, and Homo sapiens were not like in the form that we are today as the most, and we can see that difference, and I'll talk about it in evolution chapter, with respect to the cranial capacity, the size of our brain for humans. Okay, so when we get something, we try to sequence. If we get any soft, for the DNA, we will not get the entire DNA preserved. But let's say we get bones, and especially the bigger bones, they can have soft tissues trapped inside them, which just gets fossilized and preserved. You understand? Because whatever happen will happen to the hard bone outside. Inside, things can get preserved. For example, if a mosquito you all must have seen Jurassic Park, right? I do not need to tell you about that. So if a mosquito gets preserved in an amber, then it can just stay there because the resin will not allow that mosquito to degrade or microbial degradation or attacks, right? So it's kind of preserved in time. You understand what I'm talking about? So basically that was the scientific concept of that movie, that they took the blood out of that mosquito and that mosquito was sucking the blood of dinosaurs. So the blood which actually was present in the brood pouch of mosquito was a dinosaur's blood. And if it is a dinosaur's blood, it just may having blood cells. And if we can get the DNA of those from those cells, we can just make dinosaurs. Yeah. So that was the fiction, science fiction part.
So is that actually possible in real life? Just asking out of curiosity.
Yes, it is. Given that you get a DNA intact. But there are some more complexities to it, that where will you develop that? Like you need a womb, and the closest relatives of dinosaurs are reptiles, right? So dinosaurs were reptiles, then birds evolved from those reptiles. There is, I don't know if it is in class 11th or 12th, but the cover page of NCERT used to be a bird called Archaeopteryx. Do you have the brown cover? I remember that it was a brown cover. Your biology books have brown cover?
No, so it's green.
Green, okay. So it's 11th class which has brown cover, right? But evolution comes in 12th, okay. Anyways, but it is either 11th or 12th. The cover page, I don't know if they have changed it or what, but it used to be of the Archaeopteryx. It is a dinosaur, a bird. Basically, it's a dinosaur that was a connecting link between a reptile and a bird. This is the first evidence that tells us that birds evolved from reptiles. And how can we say that? Because the feature for a bird is beak. Birds have beak, right? But reptiles have teeth. They have a snout and teeth inside. Yes or no? Any reptile, like lizards. So they have teeth in a snout. But birds do not have teeth. Now this Archaeopteryx had a beak and the teeth were inside the beak. So it looked like a bird, but when it will open the mouth, there will be teeth inside. Second, it had feathers. Reptiles do not have feather on their body, right? Archaeopteryx had feathers on like some parts of the body, and on some parts it had scales. Now reptiles have scales, right? If you see snakes, they have scales. You understand what I'm talking about? So it had mixed features of both. So this was, even if you can go to internet, go and try to figure out what is Archaeopteryx. Okay, it is a connecting link. Yeah, so it's possible, but it takes a lot of formations and a lot of things to do. Actually, very interestingly, out of curiosity, when you're asking, Atif, let me tell you one thing. Recently, a group from, I think it was a Chinese group of scientists, like the name on the paper were some Chinese scientists, but I don't know if the research was done in China or in US, but they managed to develop a chicken embryo with teeth inside the beak. This tells that they can unlock certain abilities that their predecessors used to have. So chickens are birds, right? But they are still the closest cousin to dinosaurs. It's a different thing that when you look at a chicken, you do not get frightened. And in fact, sometimes it's on our tables, cooked and roasted. On the other hand, dinosaurs were ferocious, but yes, they are close cousins. And you can do that. And also, one more thing has been, it's a very interesting thing that, that tails, I told you in the last class that all the embryos from all different organisms, they have a tail bud when they develop. So all embryos look very similar, regardless of the fact whether it is a chicken embryo or a human embryo or a mouse embryo or whatever. They may be smaller or bigger in size when they are growing, but during the initial stages, did I tell you that they have a big head bud and a body with a tail bud, right? Did I tell you this? So all embryos look like this to begin with.
Yes.
And they also have gill slits. Even human embryos have gill slits, which later on do not develop and vanish because we do not need gills anymore. But this tail bud in animals which have tail keeps developing. So in humans, just the vertebral column develops. And at the very end, the tail does not develop. And the last bone of the vertebral column in humans is called coccyx. If you look at a skeleton, and if you look at the last bone is like this of a human vertebral column. You understand what I'm talking about? So the last bone is slightly bent like this, and it is called coccyx. This is a biological proof of a rudimentary bone that was the precursor of the tail bones. When not our ancestors, so our ancestors never had tail, but when humans diverged from other organisms, like when chimpanzees and humans diverged, chimpanzees also are tailless. So that's why they are the closest to us. But monkeys have tail, lemurs have tail, you understand? Only apes do not have tails. Apes are human-like. We call them humanoid or hominids. So apes, which are the advanced primates like orangutans, gorillas, and chimpanzees, being our closest, do not have tails. But monkeys, lemurs, other monkeys, they have tails. So they started living in a habitat where tail was still required. We started developing in grassland. So that's why, Africa is known as the cradle of the human civilization. Africa is where Homo sapiens evolved from. That's called the epicenter of human civilization with respect to biological connotations. Because the region from where the earliest fossils of humans are found is a grassland. It is grassland area. So we do not clearly, any organism evolving there for millions of years will not require a tail to do good. But this is just, you can still not believe me, and in science, whatever is not backed by proof is just a theory or a hypothesis. So just having a coccyx at the end of our vertebral column does not say that we ever diverge from something. It can just be like, yes, the bone is shaped like that. Don't make stories around it. So science also does that. Science criticizes itself to find more proof, able to prove it. Now, very recently, last year, a group showed that there is a gene that actually is responsible for the development of tail in an embryo. And if that gene is active during the embryonic development, then the baby that will keep developing, the embryo and the fetus that will keep developing a tail if that gene is active and is present. Now, if we were something entirely different, came from an alien planet or like something of a very different species which have no connection with any other species on the planet, then we will not have that gene, right? Yes or no?
Yes.
Yes.
But if I say that the gene that is responsible for tail development, and the experiment was done in mouse, so mouse have tail, right? Yes or no?
Yes.
If I cut the tail of a mouse, now let's just discuss this thing, because that will prime you for the next chapter, which is evolution. So if a mouse have a tail, and if I cut the tail of two mouse, so the mouse are tailless, and then I reproduce both of them, in the next generation, will the mouse have tail? What do you think, Zed?
Yes.
Yes, why? Parents didn't have tail before they reproduced. So I cut the tail before they reproduce. So will they have tail in the next generation? Will the progeny have tail? Atif? Zed says yes. What do you think, Atif?
Yes.
Yes, why? You both saying yes means that
you need to remove the gene for the tail to not develop.
Exactly. Perfect. You need to remove the gene because the gene is there. So it will be there in the sperms. It's the same like if a man in an accident loses a leg or a hand, it's not that the progenies will be born legless or limbless, right? So gene gives rise to these features. True. So they shortlisted the candidates and they figured out which gene is specifically required for tail development. And when you do not allow, like when you knock out that gene or you remove that gene or you disrupt that gene or you make it non-functional, then even when two tailed mouse will reproduce, and from the zygote, after reproduction, after fertilization, from the zygote, you removed that gene, then the mouse will be tailless, right?
Yes, sir.
Or you can do it in the gametes and then make them fertilize together, then the progeny will be tailless.
So this type of experiments on mouse, is it ethical?
In mouse, yes. And it is in fact done in the mouse. It is also, so ethical issues comes for humans. For mouse and other organisms, there are other issues like that also includes ethical issues for the animal. But if you can justify the reason for that project, why are you doing that experiment, what will it lead to finding, and how is it beneficial to humanity, then yes, they have to be scrutinized, they have to be allowed by an authority. You cannot just keep making mutant mouse. And every time we make an organism, we do an experiment on an organism, that organism have to be sacrificed, have to be killed, because we cannot leave that organism after a experiment on it in the wild. You understand why?
It can lead to other progenies.
Yes, it can disrupt the ecosystem.
Yeah.
Whatever we have done is an artificial change, and we do not want it to go and merge with the wild type and basically contaminate the ecosystem. So yes, so they found it in mouse and they figured out that the gene is also present in human, but is just inactive because it has some. So let's say this is the gene. This is the gene that they found in mouse. Okay, and it was active. That's why mouse have tail. So this was the promoter region, and every gene ends with a terminator region. You understand? Now the same gene is this is present in human, but humans don't have tail, right? So clearly the gene is not active. Yes.
Yes.
Have you heard of these rare incidences where sometimes human babies are born with a small tail?
No.
Yes, sir.
There are very few. You have heard of it, Atik? Very
Yes, sir.
small elongation of the vertebral column. So in human, the gene has some things in between, which actually makes the gene. So when you now, it's the same thing, but in between there are some random sequences inserted. So these sequences do not allow this gene to get transcribed and become a protein that is functional and that can lead to development of the tail in humans. But the fact that the sequence in a semi-conserved manner is present in human tells us and is proof enough that once in the past, our ancestors like diverged from some organisms who were tailed, who had tail, right? Now, when we do not need and we do not make tails, the rudimentary coccyx is a proof of it. And even genetically, the proof is found. It was recently last year's research paper. Interesting, right?
Yes.
So evolution tells us that we have to be humble. We are not very, very special, a creation on the planet. We just belong to a ecosystem, a chain, an evolutionary chain, and we do not own the planet. We are just one of the animal species living on the planet, and we are the most intelligent, of course. And if we are intelligent, if we have powers, then with great powers come great responsibilities, that is to preserve the planet, try to not harm it, but currently we are doing the opposite. But okay, that's fine. Probably you will understand and go back and tell to other people. And education can bring the change. So now let's finish this thing. Write down. You have written all of you have written this part. So this is a question. Remember that this can come. So prepare it like a question. From Human Genome Project, the most common question is what were the goals? Okay, and write down. The project lasted for 13 years on paper. So it was a 13-year project on paper, 1990 to 2003. It was a 13-year project. Which got completed in April 2003. But the thing that Atif said, just to make it clear, in May 2021, the last bit of some gap filling sequences were done. But it was still 2021. Okay, Atif, I don't know if you are, where are you reading 2022? But it was finished, the fund according to the funding on paper, in 2003. The Y chromosome and some, yes, it got finished in 2006, and finally some gaps also got finished in 2021. But for your entrances and for your answers, you have to only refer to NCERT. Okay, even if in the question paper there is an option 2006 and 2021 and 2003, you have to choose 2003 because entrances are from NCERT. Okay, NCERT is outdated at many points. NCERT is outdated, the knowledge is outdated, but still you have to stick to it. So I'm telling you what is correct, but what you have to stick to. Understood, everyone? 1990 to 2003 is what you have to choose. Yes, no? Okay, cool. Now write down who funded it. It was funded by United States Department of Energy. This was a neat question as well. It was funded by United States Department of Energy and NIH, National Institute of Health, again United States. It's called NIH. Sometimes it comes as NIH. The full form is National Institute of Health. Okay. Apart from these two, so these two were the primary funders, primary funding agencies. Apart from that, there were many countries like United Kingdom, Japan, France, Germany, and China. Okay. Now read this part from NCERT. Okay. And you will find some names of the techniques which were used in this project. So let me tell you in brief. You don't have to go into the details of these. So first was to figure out, now if you have to figure out all the genes that code for RNA, there is one way you can directly do it through the RNA. Yes or no? If you can find out all the RNAs that are expressed, that are transcribed, then it is clear that RNA is transcribed from a gene only, right?
Yes.
So identifying genes become easier if you can identify it through RNAs. That is what I'm saying. Yes or no?
Yes.
Yes, write down. One first approach was to focus on identifying genes that are expressed as RNAs. Okay. Now this is done through a technique called EST, expressed sequence tags. I don't have to go into the details of this, but remember EST was used. Expressed sequence tags were used to identify genes that are expressed as RNAs. And the second was, it is called a more blind approach, where you do not know the target, but you simply sequence the whole genome that contained all the coding and non-coding sequence. Coding is that codes for protein, non-coding is that do not codes for protein. Okay. And later you have to arrange them in the sequence with functions. Okay. So that is called sequence annotation. Second approach was sequencing whole genome and do this technique, this approach, sequence annotation. Just remember these two words for your entrance. You understood sequence annotation, where you do not have a target like genes that are expressed in the form of RNA, where you can put a tag and you can just focus on the express sequence tags. This is sequencing the whole genome, which includes both coding and non-coding. Coding means protein coding. Non-coding region, and then do sequence annotation to arrange it. Now, how was it done? To do it, you have to just remove the total DNA from the cell and then convert it into fragments, because sequencing a big DNA, which is a very long polymer, it is not possible. It is technically limited. Okay. So you take all the DNA, you make fragments, and when you are making fragments, you know where you are cutting the DNA at. So when you will make random fragments, you do not know where you are cutting the DNA at. So you have to make random fragments, and then all these random fragments, when they will again, you will try to arrange it, you will see that some of these fragments always will have a overlapping region. Yes or no? Because it's random. So fragments will have overlapping regions. And on the basis of that overlapping region, you can say that which fragment came first and which fragment came afterwards. So this is a complicated technique, but I'll tell you a little bit about overlapping fragments in DNA fingerprinting. But that's not exactly how sequencing was done, but similar. Okay. So write down the steps. If this can, I have seen this as a question once. Steps of sequence, DNA sequence. So first, total DNA was isolated or extracted. from a cell. Second, DNA was converted into random fragments then these fragments were cloned in a host in another cells. Basically, it can be bacterial cell or any other cell. Using specialized vector. Why was this cloning done? We basically wanted to amplify. So you can take DNA from one human cell, but you need to amplify it. So for that, you have to clone it in a other host. But because they are DNA fragments, they can technically be cloned in any host. Okay. They have no functional use in that host. So cloning resulted in amplification. Amplification of each fragment. And then once you have amplified each fragment, you can sequence. Sequence the fragments. And in NEET, one question that have come about this host, what hosts were used? So about the host, I'm adding one more point here. You can write down. Commonly used hosts were one was bacterial cell and the other was yeast. Yeast is a unicellular fungi, okay? Unicellular eukaryote. Bacteria is a unicellular prokaryote. And if it was done in bacteria, the vector that was used, so in both there will be different vectors. Vector is what carries our fragment. So remember in biology, vector is something that carries that piece of information, okay? Or that carries something from one system to another. For example, mosquito is a vector that carries plasmodium into itself and then transfer it to us so that we can get we get malaria as a disease, okay? So in bacteria, this vector was for the fragment of DNA. What carries that fragment of DNA? So in this case, vector was bacterial artificial chromosome. Bacterial artificial chromosome. Also known as BAC. 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 Okay. And in yeast, the vector was East artificial chromosome. Yeah. Simple, not difficult to remember. YAC. Okay. And this sequencing was done using a method. So this sequencing was done using a method developed by Sanger. It is still used. We call it Sanger sequencing. Frederick Sanger. So it is known as Sanger sequencing. Is it clear?
Yes.
And after that, the 7th step was, once you have sequenced the fragment, these sequences were arranged on the basis of some overlapping fragments. And this was not possible manually, so it was done through computers. Okay. So overall, it was 1990 to 2003. Okay. 13-year long project. But as I told you, in May 2006, the last part got sequenced, which was a chunk from the chromosome number 1. Now, can you see that chromosome 1, you must have thought that it is the first chromosome, right? So it must have completed the first. But some portions of chromosome 1 got completed in 2006. And some gap sequences were filled in very recently in 2021, which is just. But when it comes to how long was the project, you have to choose 2003. If the question, so read the question. If the question asks when was the last bit got completed, it was in 2006, according to your syllabus. Okay.
Yes.
Very good. So I think one more question, just like I have this question, that what were the steps of DNA sequencing? Remember this? Another question was, describe the goals of Human Genome Project. There's one more question that they ask. What were the salient features? Now in salient features, we talk about result and inferences. Remember the question, what were the salient features of double helix structure? Yes?
Yes.
Yeah, it's similar. So write down the question. Describe the salient features of human genome. It's not about the project now. It is what we got out as a result. Describe the salient features of, what did we learn about human genome from this mega experiment? First thing that we learn, that human genome contains 3.16 billion base pairs. approximately 3.16 billion base pairs. Second, about the genes, we learned that the total number of genes was around 25 to 28,000. And third, the average gene, like the average length. Average gene contains around 3,000 base pairs or bases. And also in this Ames asked once, what is the largest gene? So this is the average gene that contains 3,000 bases. Okay, there are more genes smaller than this. There are also genes that are bigger. Okay, the largest known human gene is dystrophin. Largest human gene, according to NCERT, is dystrophin. Human dystrophin gene, it is called HDG. Dystrophin. You want to know about it? So this gene recently, like not recently, but it was found that this is so long a gene. You know the speed of DNA replication for the DNA polymerase, the speed was around 2,000 bases, right? Per second.
Yes, sir.
Yes. So RNA polymerase also must not be very, very slow. It must be somewhere around it, right? Not remembering the exact speed of it. But these two molecules are very, very efficient and very, very fast. Still, it requires around 16 hours for this gene to be transcribed. Continuous work for 16 hours, a 16-hour long shift done by the RNA polymerase to transcribe dystrophin gene, because largest human gene contains around 2.4 million bases. So it's not like 3,000 bases is just an average, okay, given in NCERT. Dystrophin has 2.4 million bases. And it takes 16 hours to get transcribed. Okay. And also, what was the most interesting thing found was that 99.9% of bases, of nucleotide bases or sequence, is same in all humans. 0.1% is what makes all the difference in appearance, color, everything. Okay. And still, see, when you are finding genes, I told you what is the criteria to find genes, Atif? How do we find genes? Simple. You look at the start codon and the end codon. If it has a starting point and an end point, it is a gene, no? But just finding out a gene does not tell us what that gene does. So the function. So more than the function for more than 50% of the discovered genes are unknown. %50 plus genes, or write down the function for over 50% genes is unknown. We don't know what they do. Every day you will find studies telling that this gene is responsible for this, a new gene has been found to playing a crucial role in this and that, etc. And one more thing that I told you, how much of the genome codes for proteins? How much of the genome is cistronic? Less than 2%, 1 point something percent. So less than 2% of genome codes for proteins or is cistronic. You understand? Now you understand cistron, right? Atif,
Yes,
Hiba,
sir.
Zayed, Rifat. And they found that mostly our genome is full of repeating sequences. So some sequences that do not do anything, like as for now, we know that they are just filling up the space. So these repetitive sequences are a very large portion of the genome. Okay. So write down repeated sequences, which some repeated sequences hundreds to 1,000 times they repeat in sequence, in series. Make up a very large portion of the human genome. Okay. And let me tell you that it was thought that they are just waste and junk, they do not do anything, but as studies are progressing, we are figuring out that actually they do not code directly, so they do not have a direct coding function, but they're important for chromosomal structure, dynamics, and evolution. You know, today only I was reading a research paper which shed some light. It was a study conducted in people who actually consume too much of alcohol. So did I tell you about the telomeric chromosome? Telomeres are large repeating parts at the end of the chromosome. So let's say in a chromosome, it's a sequence. And if this is where the last gene ends, but even after that, there will be a long sequence of repeated nitrogenous bases, which is called telomeres. And this length is very, very important to protect the chromosome from damage, because whatever comes and starts attacking the chromosome, it attacks from one or the other end, right? It is very difficult to attack a chromosome sequence from the center or in the middle. So over time, as you age, so this is when you are young. When you are old, your telomeres get shortened. Interesting, right? So in an old person, this telomere gets shortened because this much has degraded. And a short telomere has also been found to be in patients who during the old age related diseases. There are some diseases that are more prone in the old age, like heart diseases, some kind of a cancers or tumors. So in those old age related diseases, it has been seen that chromosomes have short telomeres. Okay, so these repeated things or these sequences also protect. And people who consume too much of alcohol age faster, their telomeres degrade faster, but there has to be a threshold. So beyond that threshold, it starts affecting. Beneath that threshold, the other study has suggested that alcohol in a very controlled, just like chocolate, in a controlled consumption is actually very, very healthy for your cells metabolism and for energy production. But it is addictive, a substance. So once you get on it, it becomes difficult for you to figure out like where to stop. So if someone can manage to consume alcohol in just limited quantity throughout the life, he may benefit from it, he or she. But if it goes beyond a dose, then it has been shown that telomere starts depleting. Understood? Today we talked about a lot of research-based stuff, but I think that's what Human Genome Project opens as a Pandora box. Okay. So repetitive sequence is clear. They're not completely waste things. They actually help in chromosome structure, dynamics, and evolution. And you should know that chromosome number 1 is the largest chromosome with the most number of genes. It has somewhere around 3,000 genes approximately on one chromosome. And the smallest is Y chromosome. Those are all genes. They are not protein coding, all included. Protein coding are even less. So Y chromosome is the smallest, and it has very few genes, around 230 genes, and even less protein coding. Okay.
Yes.
Now, someone was telling in the last class that what are SNPs, SNPs. Someone was giving me the full form of it. I asked you to go and read SNPs, right? Did you go back and read SNPs?
Yes, sir.
Yes, SNPs and VNTRs. What was SNP, Zed?
So single nucleotide polymorphism.
Yes. As the name suggests, single nucleotide, which means single base, right?
Yes.
Polymorphism. So it has been found after the Human Genome Project that there are around 1.4 million locations in human genome. 1.4 million locations in 3 billion base pairs is not much, right? It is very less. So, but still, there are 1.4 million locations where a single base DNA difference occurs. So if I compare between my and Zad's, let's say, let me take one gene, which is working the same in me and in Zad as well, but there is possibility that we may find that there is a SNP in that gene. And that SNP will actually help me to figure out who is Zad and which is Zad's DNA and which is my DNA. You understand? So that's called single nucleotide polymorphism. So there can be single means one, nucleotide is nucleotide. So single nucleotide. Polymorphism means, poly means many, morphism means the state of appearance. So at a single base, there can be multiple different ways possible. So in my case, it can be A. In case of Z, it can be a T. But the gene will be same, the function will be same, nothing will be different. Okay, but if you compare my and Z's genome for that particular region, you will find that this is different. Just a single base is different. So at 1.4 million locations, where a single base DNA differences are there. Understood, everyone? That is called SNPs. SNPs are very important tools and a handy tool to do DNA fingerprinting. So DNA differences. Occur in humans. Okay, everyone?
Yes.
Okay, so this information is important for tracing human history and also to do like specific mapping of ancestors, etc. Okay, so the last part in the chapter is DNA fingerprinting, where I have to tell you about a technique, how basically it's done with the help of a diagram. How much time do we have? What's the time? We just have 10 minutes. Okay, we'll probably not be able to finish it in 10 minutes today. So that's the last part of this chapter that I'll finish in the next class, and I'll start with evolution. A lot of things regarding evolution we discussed in today's class. Okay, so remember, let me tell you what things. So the last thing that we'll be doing, is this clear? Is human genome project clear? Everything? Just go from top to bottom and see if everything is clear to you. We have done the goals, we have done the techniques, steps, and salient features. Okay. Now, DNA fingerprinting. Let me prime you to this thing, because this is in detail. This is also asked in detail in entrances. So what do we mean by fingerprinting? What does the word DNA fingerprinting tells you? What is a fingerprint? Then fingerprint.
So it is a map, we can say. It is a DNA. No.
I'm asking, what is fingerprint?
So how can I explain it?
Arey, you give your fingerprints everywhere, no?
Yeah.
For your identification, you have to put a thumb impression. What is it?
It is a design on the finger.
Yes, a design on the finger, right? How is that design created? Who's the designer? Can you get your fingerprint redesigned if you don't like it?
No, sir.
Let's say this is a thumb on the opposite side. So on your thumb, you have some designs like this, right?
Yes.
But what is this design? Where does it come from? It is a unique thing, right? You know, every human has a different fingerprint, and that's what is the important thing about it. And this is just it's very randomly you get these fingerprints when you are growing as a baby, as a result of folds and pressures that come on your skin and how it grows. You understand? So DNA fingerprints are unique and different. Let me ask you a question. Can identical twins have same fingerprints?
No, sir.
No. But they are identical twins, no? They have same genetic information.
Yes.
So can they have same fingerprint? Because identical twin have same DNA fingerprint. That's where the confusion comes. If someone is an identical twin of another person, then basically they came from the same zygote. I think Atif popped out. Yes, Atif, can you hear me? Atif, am I audible to you?
Yes, sir. I left the class by mistake. Can you repeat it?
Yeah, I'm asking, we are talking about DNA fingerprinting, but I ask, what is fingerprinting? Yeah, or what is a fingerprint? So we all have fingerprints on our fingers, right? Which we provide as identification proofs. We give it as biometrics, right? For our passports and things. Because every human has a different fingerprint. My question is, do identical twins have same fingerprint?
No.
No. Do identical twins have same DNA fingerprint? Okay. Let me leave you with this question. Read about this question. If yes, why? If no, why? And the second part of this question is, can identical twins have same fingerprint? And second part is, can they have same DNA fingerprint? Just try to know this answer. And even if you don't understand the logic, that we will discuss in the next class. And I'll finish with this topic and we'll start evolution in the next class. Okay.
Yes.
Okay, everyone. Any questions for today? See, go through NCERT, read every line from your NCERT regarding these two topics, DNA fingerprinting and human genome project, and then come in the next class. Okay.
Yes, sir.
Yes.
Okay, then. If you have no doubts, then I declare the class. Take care. Good night, everyone. See you in the next class. Bye-bye.